Good day, everybody. I am very honored today to receive the ASM Award for Environmental Research in Microbiology. This presentation is meant to describe how team science has given us excellent access to various projects in environmental systems biology. Environmental systems biology starts at ecosystem level and goes down to the molecular level, resulting in models that provide increasing complexity up to the ecosystem level. Systems biology, the whole is greater than the sum of its parts. Multiple lines of evidence, though, are needed across all levels. System biology, as I said, requires multiple levels of evidence across all various levels. We are fortunate to have a plethora of new methods for each level to provide the evidence we need to test our hypotheses and to build the models we need at various levels to realize our goals of systems modeling. None of the examples that I am about to present would have been possible without large interdisciplinary teams that take advantage of all of these methods at the ecosystem level, the community level, the population level, the cell level, protein, lipid, and metabolite level, and nucleic acids themselves. My friend Gary Saylor, one of the previous recipients of this same award, and I published a review in Environmental Systems Biology and Field Test Plans in the ASM Manual of Environmental Microbiology in 2016. Basically, we showed that there needed to be a plan where you had hypotheses to be tested, defined boundaries of the environmental system, measurements to be made, sampling protocols, holding time, storage, etc., and then resources needed, costs, equipment, people, time, and a field test plan, including permits, hazardous sampling plans and safety plans, protocols, responsibilities, budgets, priorities, databases, and data management, and science of opportunity. You would then go to mobilization, and finally to implementation, where then you would provide feedback to the field test plan and where it needed to be modified. Finally, you would demobilize and then do reports and publications. So that's the basic scenario that we need to go through. Now, quite often I need field personnel out in the field to take care of anything that might happen. And so I use this list quite often to say, okay, we probably are going to require a geologist and drilling experiments. And basically, those people are asking, where's the clay? Hydrologist and environmental engineer, where's the water? Microbiologist, where's the bugs and or genes? Analytical chemist, two of them. One for where's the contaminants, where's the non-contaminants? An engineer, construction, mechanical, where's the equipment? Business manager, where's the money? A modeler statistician, where's the data and data management? And then on call, electrician, plumbers, electronic repair, computer programmers, and it could be food or whatever else you need, but where's the problem? Now, one of the early studies that I was involved in was at the Savannah River site and the Savannah River integrated demonstration of in situ bioremediation. And in fact, we had both analytical and process control. We had ORNL doing various activities and having separate contracts with the University of Tennessee, University of Minnesota for nucleic acids, phospholipids, etc. And then the Savannah River Technology Center, which is now the Savannah River National Laboratory. They basically had plans with or contracts with the University of North Carolina and with EPA and Mantech and Stanford University. Pacific Northwest Laboratory, they had contracts with Washington State University, and they were doing enrichments, fungi, and actinomycetes. We also had field site operations with Ecova, Westinghouse Savannah River, and Battelle Savannah River, and the Gas Research Institute and Radium. On the process control side, we had ORNL with PCE groundwater bioreactor and a co-metabolic bioreactor from the U.S. Air Force. SRTC had trickle filter bioreactors from the Air Force and trickle filter bioreactors from other agencies and fluidized bed from Gas Research Institute, Michigan Biotech, and Virex and Radian, and gas phase bioreactors with Ecova and soil column simulations. INEL had a soil column bioreactor that they operated with us, and the Los Alamos National Laboratory operated biodegradation modeling. What this particular project showed is that with methane oxidizing bacteria, which are ubiquitous in nature and can co-metabolically degrade over 1,000 compounds, including toxic chemicals, including trichloroethylene. And because we had a trichloroethylene problem, we actually patented this process with the U.S. Department of Energy. And this represented the first full-scale field demonstration for DOE in 1994 at Savannah Riverside. You can see here actually what happened in terms of a real-time 3D spatial and temporal model of actual data, which allowed us to demonstrate how methane biostimulation worked and allowed the companies that bought a license from DOE how they could use it to clean up sites in the US and abroad. Here you see the two horizontal wells, and in fact, one above the water table and the one below the water table, and this area of clay lenses that was associated with the TCE. And here you can see with that blue layer, the TCE that's associated with some of that clay layers. And then this brown layer that grows is actually the methanotroph densities and how those change through time. And remember, this is real data. And then what I did is to go in and look at the chloride concentration and how that had changed in the project, and could show that there was chloride where it should be above and below the clay layers. And in fact, that showed that the trichloroethylene was degrading completely to CO2 and chloride. We also related, and this is sort of related to the integrated demonstration project and other DOE projects on the deep subsurface. A team headed by Indiana and Princeton started a project funded by NASA. Our papers received some attention from Ripley's, believe it or not, and though they did not let the facts get in the way of a good story, in fact, illustrated the popularity of some of the stuff that we were doing. And this is just two of the science papers that we have on this. And the reason NASA was interested in this is, of course, because it potentially could represent life on or under Mars. And despite knowing most of the chemical conditions, we could not grow this bacteria that we found, the Sulfurudus adaxiator, but it was 94% of the 16S and 99.9% of the metagenomics. And you see the large group of people here that were involved. So this could be an actual biomarker for extraterrestrial life. It's 8,000 feet in the fractures of South Africa, and it's been isolated for over 1 million years. The radiolysis we showed would create hydrogen peroxide bicarbonate, which in turn affects perennial rock material containing ferrous sulfide, which would release sulfate and phosphate and dissolution of calcite, which releases ammonium, giving the cell all the things necessary for growth and activity. So it was living down there in a quiescent state, but we termed it life in the slow lane because in looking at the turnover times of isotopic components in compounds that were found in the cells, basically it was occurring from 40 to 400 years, hence life in the slow lane. Well, Deepwater Horizon came along and we stepped up for that. And in fact, here's a couple of papers that we've published on this. And basically this rig caught on fire from methane, collapsed into the ocean down to 1,500 meters. And then there was two plumes, one going up to the surface and one going out more or less horizontally in a cloud of oil droplets, 10 to 60 microns wide. There was also, BP was putting in Corexit 9500, a dispersant to help create smaller droplets and help disperse it more widely and perhaps increase the biodegradation rate. So the marine oil spill systems biology approach basically showed how we could do hydrocarbon stable isotopes, nutrients, physical and chemical analyses, lipids, phospholipid fatty acid analysis, metatranscriptomics. This was done by the Joint Genome Institute. Isolation and phenotypic characterization of cell counts, cloning and sequencing, 454 pirate tag by JGI, phyla chip by us at Berkeley Lab, geotip by the University of Oklahoma, metagenomics by JGI, and then single cell sequencing with JGI, and at the Advanced Light Source, synchrotron imaging for various components. What we found was that in the uncontaminated part of the plume, that less than 3% were the oceanus spiralis, which we found in 80% of the proximal part of the plume and 93% of the distal part of the plume. And in fact, at the distal part of the plume, the oil was, by most methods, was undetectable, but it was still showing a distinctive case of it being able to be detected. We also looked at the distribution of organisms and the succession in the microbial community. We saw the oceanus spiralis come up rapidly at first, and Pseudomonas and Coelia, and then gradually it dropped off to, once the well was shut in and there's no more oil going into the deep, to these other organisms which could degrade the components that were left after the oceanus spiralis, Pseudomonas, and some of those ate the good stuff. We showed that by our single cell sequencing that this uncultured oceanus spiralis had genes for chemotaxis, nutrient acquisition, cyclohexane degradation. It did beta oxidation and basically had various mechanisms for putting critical nutrient components both into the cell and out of the cell. And it also had chemotaxis to oil itself. Well, when our paper came out in Science, the Houston Chronicle printed this cartoon. Sometimes your team will need thick skin to weather public reaction. It clearly got some excitement, and of course, tongue-in-cheek, a huge oil-eating bacteria just ate Earl. Science of Opportunity showed us that, in fact, BP had licenses offshore Angola, offshore Caspian Sea, offshore Central Mediterranean, in the Eastern Mediterranean, Great Australian Bight. And they also wanted to see if, in fact, what the oil biodegradation rate would potentially be. And this is true mineralization to CO2. And you can see none of them performed as well as the Gulf of Mexico. And the Corexit 9500 worked the best in the Gulf of Mexico, clearly. The Enigma, or ecosystems and networks integrated with genes and molecular assemblies, is one of our big projects that we've had going on for a long, long time. It has an aim of subsurface observatory, an environmental atlas, and environmental simulations. And we've had this going on for several years. It is multi-institutional consortium of universities and national laboratories that is funded by the United States Department of Energy. The constraints on the community composition activity, and we're focused principally on Oak Ridge Reservation and on the Bear Creek Valley, and showing the S3 ponds, 4 ponds where they dumped mixed waste, which included nitric acid, uranium, and a variety of other metals. That went on for over 30 years, and then they neutralized it and capped it. This included it's now an asphalt parking lot, but it continues to leak material along the sides. And we, in fact, have recorded the highest concentrations of nitrate in groundwater anywhere in the world here. It gets as high in a couple of wells as 11,000 parts per million. So our field test plan, the first thing we wanted to do was a 100-well survey. There's over 800 wells there. And so we basically keyed down on the 100 best wells that were mutually exclusive, and then set up a plan to identify staffing, leads, hypotheses to be tested, the number of effective replicates, the minimum significant sample size, methods comparisons, and various things like that. And this took over 2 years. Finally resulting in publications and other campaigns, other techniques and protocols, and future work. The bacterial 16S sequence data using our slime model that was developed with the folks at MIT predicted with the 16S community structure 18 different highly significant correlations, including pH, uranium, cadmium, potassium, magnesium, etc. And then there were 8 significant parameters also, aluminum, chloride, and that sort of thing. But there was 12 that weren't significant at all. Science of opportunity. So because we had this data, and we had a lot of data from Deepwater Horizon, in this same paper in MBio, you can see where we used that data that we had on the microbial community structure, looking at the sensitivity and specificity to predict the oil. And it predicted the oil almost perfectly, even better than it predicted the uranium and nitrate, which was actually quite good also. Subsequent to that, we also became part of a consortium for monitoring technology and verification with the National Nuclear Security Administration. And basically, this is a proof of principle of detecting nuclear production activity for environmental samples. That could be soil, sediment, groundwater, wastewater, and cooling towers. We're doing this in 2 different sites right now, and at the Savannah River site, and then at ORNL and Y12, basically because we have various ground sites, we have various types of contamination in Y12 that, like we just showed you, with S3 ponds, but we also have a functioning nuclear reactor, the high flux isotope reactor, which has been going since 1965. So we're looking at the environmental areas around that. Enigma has done a number of field campaigns since finishing the 100-well survey. And these include a sediment plots, both up above and below the S3 ponds, and looking at a variety of different sediment samples. We published quite a few papers on that already, but we still have a lot more to publish. We also did a geo deep dive and looking at continuous monitoring by using remote telemetry And this is the first time that the secure site at Y12 has gotten remote telemetry into a site that we could then monitor. Everybody in Enigma can monitor the particular wells that we have hooked up to this device, and also the meteorology, so the weather. And then we did integrated sampling, which basically showed us that what we thought was our background site was too far away. We also showed that we needed to do more frequent sampling than every other week. So we then went to 27-1 survey, where we monitored 27 wells, 4 days a week for 5 months in 3 different areas. And and we also did real-time nitrous oxide analysis there. And those papers are just coming out now. We also then did the cone penetrometer to determine the lithology of the subsurface in Area 3 for the installation of our potential subsurface observatory. We had 7 labs analyze 103 parameters for 292 samples that we shipped to collaborators. In the cone penetrometer, we can actually see up here in the S3 pond source material that we had all these different pushes. So we had a 3-dimensional structure. And the hydraulic, and we could push at and get real-time data at 2 centimeters per second. So this allowed us to do all of this sampling in 16 days. The hydraulic conductivity has a tortuous path at this site because due to the geological strata being exposed to the surface in a tilted plane and then weathered, and with the sediment laid down by the river delta. So as you can see here, it's quite tortuous, and it actually goes down dip, so it actually goes deeper at the south end of the site. We could also see nitrate analyses here, and this, of course, is one of the hot sites where we see 10,000 parts per million. On our subsurface observatory, we're setting this up so that we have 2 vadozone piezometer and 2 variable saturated zone wells and a saturated zone sampling point in each well of 9 wells in a cluster. So we can measure flow going across and in patterns, and then up and down gradient. And we can also use this to test different movements, how fast things move, and that sort of thing. It allows us to see exactly where we need to put those wells to make sure they have connection to down gradient wells. And you can see here the left series of 3 versus the middle series of 3 and the right series of 3. Hazen Lab and people, this is people that we have in my group for the last 2 years. I must say that they took a vested interest in this presentation. And as always, we took advantage of the pandemic for Science of Opportunity early last year by monitoring all student housing on the UTK campus in wastewater weekly for SARS-CoV-2. Unfortunately, we don't have enough time to show that, but if you go to our PAUSE website, you can see some of what we did and exactly the way we did it and everything else. You can also go to the Hazen Lab website and see a lot of the papers and that sort of thing. The MTV site, of course, is ongoing too, and this is the National Nuclear Security Administration. You can go to their site there at the University of Michigan, and it has all of these universities involved, plus all of the national labs, which have National Nuclear Security Administration. Enigma team, this is clearly a large group, and we've been active for quite a long time. And it is a multi-institutional consortium of universities and the national laboratories, and funded by the U.S. Department of Energy, in particular the Office of Science, and biological and environmental research. You can contact me at any of these sites and be happy to answer any questions that you may have.